Good morning, everybody. Happy Monday. We will get started with this stuff. So we got a chunk of the way, maybe a third of the way into the respiratory tract infections. We talked about numerous types of bacteria that set the stage for causing issues. And we want to shift gears. I left you off Friday right here with influenza. We want to shift gears and think about some other players. We're going to talk about a few types of viral organisms, and then we will also talk about some fungal organisms as well as we move through here. So lots of the time, we think about things like bacteria and viral organisms that cause issues. And often the fungi go unthought of, and that's potentially sad. It's maybe an issue when we think about some of these potentials and possibilities. And, they're kind of near and dear to my heart. I'll touch on that as we move through here. But there are definitely some situations as we think about some of these fungal culprits that start to emerge in individuals that cause issues that we kind of throw our hands up and we're like, I don't know what the heck is going on. In those circumstances, you would be very wise to start thinking outside of the box a little bit and thinking about some possibilities of some crazy fungi, all right? So we'll get to those here in a minute, all right? There are all sorts of potentials when we think about virus, and influenza up here is fitting. However, I also feel like maybe we should have some coronavirus up here and talk about some details with that. But part of me is also like, I think we've all had it. So the flu is interesting in its own right. And there are some things that we can think about as bearing some similarities with our coronavirus as well. Not the same ideas, but some similar players. As we see some of these spike proteins on the flu virus, they're not the same spike proteins, but it's a similar idea that lends to the viral pathogen having the capability of making contact with respective cells and in turn, like docking, landing, and infecting, all right? And so we see these different color, different shaped as well, the H and N proteins for hemagglutinin and neuraminidase, respectively. And there is variation within these that lends to the variation of different types or strains of influenza. And we can see similarities with a plethora of other viral pathogens that have some variation within this, all right? Lending to maybe enhanced capabilities of infecting or whatever it might be, giving them a potential leg up. So there are numerous types when we're thinking as we're on the flu. And we try to produce vaccines that are effective against these specific signatures, if you will. It's kind of back to thinking about like our own molecular fingerprints and then the fingerprints of the other players. Can we detect these fingerprints adequately and mount some sort of a potential to respond, a vaccine that would in turn confer some immunity against these? The flu has a plethora of potentials, and there are various vaccines as we try to produce this, various fractions that we try to anticipate being the main player in that season's flu virus, right? And it's the result of the respective combinations of these proteins that we try to target. Usually flu vaccines are a trivalent vaccine. Sometimes there's a little bit of variation with that, but 3 different fingerprints, if you will, or signatures that we try to mount a response against. Sometimes that is effective, and other times it is not. If the strain that we see as the dominant type was not in that combination or that cocktail for the vaccine, well, you've found yourself immunized against something that you're not protected against in this case, all right? There are also other things that I think are interesting, and the flu, like a handful of others, is an RNA virus, which, as we've thought the littlest bit about some of this, and definitely focused on some other details in other courses, thinking about DNA replication or building proteins with the process of transcription and translation. When DNA is being copied, the DNA polymerases have proofreading mechanisms, and if issues happen, mismatches or something like that, for the most part, those things get corrected. But when we move over to the production of proteins, or we're talking RNA viruses, the RNA polymerases don't have those capabilities. And so the likelihood of things changing more rapidly in an RNA-typed virus is tremendous. And we can see that with coronavirus as well, all right? And numerous types of variants that we are seeing as a result of this process of replicating these structures, and it's not getting proofread. That in turn lends to other opportunities for what's studied on the outside to vary as well, all right? Lending to differences for sure, all right? So with that said, it gives us some of these different possibilities. It also sets the stage. You could imagine that there is a potential that you may contract something like this, and maybe what you are infected with is a couple of different variants. And in turn, in their process of replicating within one of your respective cells, you could get some stuff packaged up that's a bit of both. And now maybe you've got something that's considerably more dangerous, more virulent, or maybe not, right? It truly depends on those cases, right? And then that, you know, we could think of other things like bird flu and whatever, and that really depends on the respective situation, the animals that might be getting infected that could lend to it plaguing us. Usually, before we hit measles, I'll talk bird flu for 1 second. We think about a situation where there are all sorts of viruses plaguing all sorts of different types of animals. Birds and humans, very different. If we think about our own immune responses, and cells, different. And the virus that wants to plague them can't really target our own cells. The receptors are a bit different, right? Pigs and people, they're definitely more close, right? And things can be shared pretty readily with that. Well, it also happens that pigs and birds, they can share stuff too. So bird flu can get into a pig, human flu can get into a pig. Now this pig's infected with both. There's the potential for that same sort of recombination that I just described. And as it sneezes stuff out, now it's got the potential that these viral particles can target our own cells. And now we've got something very different, maybe super virulent, all right? And that's why there's definitely concerns when we think about things like the bird flu, because it's something that really shouldn't be in us, but it's got an opportunity that it could, all right? Shifted over, measles is something that we think about with vaccines to keep this away from our little fresh hatchlings, but that's not necessarily always the case, right? So this, as we see a handful of different images, the actual virus over here through an electron micrograph, and then the rash-like appearance here we're seeing inside of the mouth. of an individual, I'm assuming not the same, these Koplik spots on the mucosa, another characteristic of measles. This is typically what we think about, raised macular rash, and then spreading. This seems like we should be thinking skin infections. We showed a little bit of this previously, right? But also respiratory, and that's why we've got these suckers up here as well. Like I said before, there's potentials to see them in multiple body sites, right? And that's why we're bringing that back. German measles, here we can see this, and it's rubeola is measles, rubella is German measles, all right? And these are, they're both types of measles, but this one's a little bit more mild, all right? It's smaller lesions within here, less intense, and these lesions not confluent. So basically individual little lesions that don't merge, which is what you would see over here, right? If we were to zoom in more on that, you would see more ink, pretty much see it on his cheek in the first place, all right? Also, vaccine with that. So our next player is chickenpox, and we can see some of these pock lesions on the skin. Here's a image of the virus. Varicella zoster is what we call it, but it is a human herpes virus. Human herpes virus 3 is where this fits if we were classifying them. We think of all sorts of viruses, and I said classify. They don't fit in a tree of life, but we do look at the DNA and the RNA, and we group them up, and we do build phylogenetic trees like that. Yes?
So I know it looks kind of similar to like hives or something like that. So what kind of a distinguishing factor for doctors to be able to identify, oh, this isn't like a common allergic reaction, this is actually chicken.
Okay. So that's a great question. And with some, some of these circumstances, visualizing chickenpox now with all sorts of vaccines, more of a rarity, right? Oftentimes thinking you're going to have more than just this on your skin that's an issue. When you get hives as a result of an allergic reaction, what you're feeling inside is considerably different than what's going on with chickenpox. You feel super sick, flu-like kind of symptoms with chickenpox, and with an allergic reaction, you don't feel those same sorts of symptoms. So there's a lot of, there might be some skin-like, looking at skin manifestations, some things that look kind of like they're overlapping, but other signs and symptoms are really going to drive you down different lanes as you're thinking about that, right?
When one of my kids was little, we walked into a treatment room for triage. She was getting a strep test or something, and there was a kid in there, and I stopped dead, and I was like, looked like he had smallpox. He had large, blistered pustules all over him. And he had gotten a hold of eggs.
Oh.
He was allergic to eggs, and just that morning. And so I was like, wait, we're not going in there. She's like, no, it's fine. It's just an allergic reaction. And I was like, okay, I believe you, I guess. But it can look, yeah.
Yeah. So it truly depends. And, symptom wise, something like that, I'm sure that kid's feeling terrible, but not the same, right? So it really it just depends on those circumstances. So here we're shifting, likely decades later in life. Here we've got an individual with shingles, and it, technically doesn't have to be, much later in life, but usually it is, all right? So the virus can, like numerous types of viruses, they can take up residence, going dormant, if you will, on some nerve cells. Herpes loves to do that. And this being one of the herpes virus, similar sorts of an idea, all right? We've talked of herpes simplex 1 and 2 with some skin stuff and potentials with that. This is a similar idea, but it's something that you might think of cold sores happening considerably more frequently, something like shingles, not, all right, or hopefully not, all right. And there are potentials, just as there are vaccines for chickenpox, later in life, you can get a vaccine for shingles. It is not the same vaccine, but similar, all right. And it in turn helps to prevent this from showing up, all right. All sorts of virus within here, and a good chunk of them we have hit. Common cold, there are numerous types of virus that cause the common cold. The symptoms are super similar, all right? And that there's issues with that, and issues with, as we see over there for vaccine, there really isn't one, because it's super difficult to have a vaccine that's effective against every single one of these and all of the tremendous variation. That's hard, all right? So you can see symptoms of runny nose and congestion, some sore throat, sneezing, headaches, muscle aches, all sorts of just common cold symptoms, all right? Rhinovirus, adenovirus, and coronaviruses, some others fit in there as well, all right? All common cold-like. They, in turn, can be considerably more severe and lend us to pandemic situations, as we see down here with SARS, which fits right within here, all right? So influenza, we saw the measles, MERS is Middle Eastern Respiratory Syndrome, SARS, all of these different close cousins, coronavirus and another coronavirus, and issues with both of these, where we were potentially thinking possible pandemic sorts of situations, and it did not progress to those levels as we have seen with other coronavirus that we're hopefully marching out and away from, all right? I sure hope so. We saw the measles as well, and shingles, and then a handful of viral pneumonias. And this is something that we would need to be differentiating between what we ended with Friday, the bacterial forms versus viral forms. Some signs and symptoms of this will overlap, and some will set it to things that clue us in other, all right? So dependent upon the situation, with something like respiratory syncytial virus and a little kid, it can be a super serious infection, all right? Otherwise, maybe more mild when we think about it, all right? Just depends. Okay. Shifting gears to some fungus. Oh man, I love them. Okay, there are all sorts of different types of fungi that we can think about causing some issues. And again, we're thinking respiratory tract, but a good chunk of these that we want to think about in this, there's more to it than just that. All right, coccidioides, histoplasma, and blastomyces. These three types of fungi we've mentioned briefly before. We showed some histoplasma. I can't remember what chapter that was, but we showed that with some bats up in an attic and whatever, all right? These fungi are what we call dimorphic fungi, and they exist in the environment or at like room temperature situations. They exist as a mold. And then if they get into a mammal, or if you ramp that temperature up to about body temperature, 37 degrees Celsius, these things will grow as yeast cells or spherules, as we see with this, but it's basically like yeast with them, right? So interesting, disturbing, and possibly terrifying, all right, as you see some of this stuff. So these dimorphic fungi are shapes shifters, going from filamentous fungi to circular yeast cells. They also have the capability of taking hold within your lungs. And then if the situation presents, if they truly can proliferate, they disseminate, and they move possibly to any place and every place within your body, all right? And then it looks like, you know, this poor gentleman here, you're molding from the inside out is really kind of how it looks, all right? But I should back that up. It's really, you know, these skin lesions, if you were to collect, it would be yeast and not mold, all right? But it sounds more gruesome to, you know, say that as you open up the refrigerator and pull out the Tupperware and here's this moldy stuff. Well, it's us as well that has that potential, and that really makes me feel a little uneasy, all right? So with these dimorphic fungi that we will think about as we move through, there's definitely potentials, again, thinking respiratory stuff, but oftentimes we may not recognize issues until we actually see some of the dissemination. And then you're like, wait a minute, right? This, you know, this chronic cough, maybe there was something else to it, right? And we didn't recognize that until this, right? Whatever it might be. It depends on the type of fungus as far as where we might be seeing these players, all right? Coccidioides is an organism that likes more of an arid type of habitat. The Southwest is a great place for this. There are areas in California where we would say this is endemic. This fungus has been called valley fever for the San Joaquin Valley, all right? And there are some pretty good windstorms, they call them the Santa Ana winds, that blow dust all over the place. And if these spores, not these guys, but if the spores from the mold have the capability of becoming airborne, that's how we think about coming in contact with them as we inhale that. And in these huge windstorms, it's got the capability of blowing this fungus for miles away from its initial source, lending to endemic situations in these areas where the fungus is just suspected it's present, all right? Whether you can actually find it or not, blowing in the dust is an issue, all right? With that being said, something like this in the southwestern areas, Arizona, New Mexico, things like that, other problems, not that, you know, Southern California isn't a potential issue as well. There are numerous individuals, the snowbirds, that will travel from Canada down to these areas, winter there, and then go back to Canada. And in turn, show up with issues that you don't see that stuff in Canada, right? It's an issue. And that's another thing, as I started this lecture today, you got to think outside of the box sometimes, right? These doctors in Canada are now like, okay, this is a problem, right? We are seeing this in these individuals because of, well, basically this lifestyle, traveling south, golfing as it's freezing cold in Canada, and exposing themselves to these pathogens and then going back, and it can take months for it to progress, maybe even years before we start seeing dissemination, before these things actually become apparent. And that's a huge problem. Yes.
And then, so for prevention, other than like not traveling, like vaccine wise, or even just like healthcare wise, or just maintenance, like what would be the best prevention?
So there are no vaccines for these things. So I think maybe recognizing the potential, right?
Yeah.
I would advise not going golfing in the, you know, in a humongous dust storm. That's probably a bad idea, right? And then, you know, with that said, there are situations as we see this with people that, I mean, it truly looks like some serious occurrences. Lots of people probably get exposed to these fungi, to a handful of spores, and their body mounts an immune response and destroys them before they ever truly can take hold and manifest. If your immune system is depleted a bit, if you're weakened, if you are more, up in age, if you're getting elderly and your immune response is weakening just by that, you don't have to have other issues. That's where you've got a greater likelihood of them coming in and actually taking hold. So that sets the stage for symptoms. You get some lung issues, further digging, not just saying, oh, it's just a cough. You probably should evaluate that and see really what's up. That's what I would think. But, like vaccines and stuff, that's not, although it could be nice, not something, at least right now, not on the radar. We talked about Legionella before. This, I think, is pretty awesome. In this circumstance, the Legionella pneumophilia is living inside of some amoeba. And this is a mechanism, and here it's from a contaminated water sample. That's oftentimes how we get infected, but we're not getting infected with the amoeba. It's the bacteria that are living inside of these guys, all right? And so kind of interesting. The amoeba, I don't think, if I remember correctly, it's kind of a symbiotic relationship. They're not like feeding on these bacteria. The bacteria are living inside of them, having a nice moist habitat inside of the amoeba. And that sets the stage for them to potentially infect us. And the Legionella respiratory stuff by itself, it's the amoeba that's slapped in there, I think, within this bit of material for why it was there and not, on Friday's material. This next one, another dimorphic fungus. This is blastomycosis, blastomyces dermatitidis. And here we can see the yeast cells. These yeast cells are really pretty large. They've got a very thick cell wall, and it's something that helps to diagnose it with like the histoplasma, also big yeast cells, as opposed to the spherules, right? So this is a big sac with these cells within it, little bit different, right? As I said, these fungi near and dear to my heart. As a graduate student, this was the organism that I studied, and I kind of felt fortunate because in Idaho, we don't see this as a pathogen. So something that can disturb you, but then you can go home and be like, but at least it's not here, right? It's okay. So this is an organism that likes more of a kind of moist environment. The Mississippi River Valley is where we would say it is endemic. So too is histoplasma, and it moves a little bit to the east, same as this, all right? So not just right around the Mississippi River, but they like those types of kind of moist habitats. And they think the moisture is something that helps to get the fungus to release the spores. And it's doing that just to continue to grow as a fungus, as a mold. But if those spores happen to get inhaled, then it sets the stage for this, all right? So foggy mornings or after it has rained could potentially have a greater likelihood for contracting this, not a dust storm, all right? A little bit different, all right? And here we can see as it's disseminated, forming the cutaneous blastomycosis, again, kind of molding from the inside out as we see that. These yeast cells would be observed in your lungs. They also would be observed on the skin if we were to make some scrapings from this lesion. And you could, one potential for verification with this would be to collect these yeast cells and to cultivate them, to grow them at both 25 degrees Celsius and 37 degrees Celsius. That will confirm that you're truly looking at a dimorphic fungus when you see mold, fuzzy mold at 25, and different yeast cells. The colonies are completely different at 37, all right? Same with all three of those. This next one, and I know we mentioned Aspergillus the littlest bit before. Aspergillus is not a dimorphic fungus, right? It is a mold. It's a monomorphic mold, meaning it's only mold, all right? So Aspergillus, the inhalation of some of these fungal spores within the lungs, a nice warm, hospitable environment, lends to the mold growing within there, causing a fungal ball, which is a big filamentous mass of the, the hyphal filaments of this fungi, creating a big mass within the lungs, all right? So aspergilloma is this as we look at it, right? And as I mentioned before, what that, like the black mold that you potentially see in a poorly ventilated bathroom or something, that's aspergillosis, aspergilla, excuse me, that can cause aspergillosis. This next one, some yeasts, as we're looking at these big spheres up in here, Pneumocystis jerebeckii, and this is again from lung. This is one that is also monomorphic. It's only yeast, right? Not filamentous fungi and then converting to yeast cells, right? And Pneumocystis is a pulmonary infection that is pretty uncommon unless we're thinking about individuals that are severely compromised. There are lots of things when we think about patients that have progressed from HIV to this status of being classified as having AIDS. At that point, your immune response is basically non-existent, and it opens up the avenue for a plethora of things that a normal individual would typically not think anything of. But an individual that can't respond, all of a sudden, it's a huge issue. And Pneumocystis is a big player. As we see issues with this, it's a huge red flag that we've got something going on and probably AIDS, right? So kind of crazy. Cryptococcus is another yeast, and this over here with some negative stain shows the huge capsule layer that you can see surrounding these guys. Cryptococcus is, we can see those, capsule layers on bacteria, and we can do some negative staining, and it looks impressive. But if you want a good one, these yeast cells are definitely ones that show capsule layers great. You can see this huge halo around them, and then the cell wall of that yeast giving it this appearance. Pretty impressive. And here we can see some more of it. This would be from a sputum specimen. We've got a handful of these yeast cells and a few of them in the process of replicating, all right? Also, another one that's pretty common when we think about AIDS patients, Cryptococcus as well. So numerous fungi that we can think about as potentials for plaguing the respiratory tract. We showed the fungus ball with Aspergillus, Aspergillus fumigatus, blastomyces, coccidiomyces, and histoplasmosis. All of these guys are the dimorphic fungi. And again, kind of some different habitats where we might find them. But mold to yeast within your lungs. And they also have the capability of dissemination, right? The mucormycosis, we didn't mention this, but it's impressive. Rhizopus and mucor are some common players that can cause these issues, all right? And both of these we may think of as a common bread mold, right? Something that you've all probably seen, right? I was teaching a non-majors biology class, and we were talking about molds and whatever, and this guy raises his hand and said he had a roommate, which makes you wonder, is he talking about himself or not, right? I have a roommate that likes to keep the bread until it gets moldy, and then he makes his sandwiches. And I was like, what? Are you serious? That's disgusting, right? And I'm like, why would he do that? He said that he says, and that's how you know there's an issue, right? He likes the taste. It gives it a tang. And I'm like, okay, all right. That is interesting, all right. I have eaten some things that are moldy because they're supposed to be. And even then, sometimes I'm just like, that's flat out too moldy. I can't eat that, right? Even though this cheese is supposed to have all of this on here, all I can do is think about, this is just mold, right? I should not be consuming this, right? And there you go, right? And it, makes me say, damn you, Costco, why did I sample that, right? Where's the drink, right? Potential issues, right? Moldy bread is not something that I'm like, oh, yeah, we should consume that, right? Probably not, all right? So these circumstances, you've got some serious issues in the first place, all right, for mucormycosis to result, all right? Severely immunocompromised issues. Otherwise, I think we would all have some big problems, because pretty sure we've all seen some moldy bread, all right? Opened up that loaf, maybe even started to make yourself a sandwich, and you're like, holy cow, there's mold on this, done deal. Hopefully it's not halfway through the sandwich when you recognize it. And sometimes that does happen, right? Issues, all right? So simply just the inhalation of a few of these spores, this fungus can get into your body, get into your brain, and can be a fatal infection in a handful of days, all right? Which is terrifying and super stinking awesome at the same time, right? Something that kills you like that, you gotta love it, right? So headache and fever, facial swelling and congestion. There might be some lesions at the back of the throat, chest pain, shortness of breath, and then last but not least, often fatal, all right? If it gets in there and it starts causing these issues, it's a problem, all right? And like I said, most of the time, none of this ever happens, all right? But if you are compromised, if that stage is set, you should use some serious caution with that, all right? So amphotericin B, there's most of these are things like amphotericin or different azole drugs that are effective against numerous types of fungi. And then the pneumocystis, we saw that just a bit ago. All right. And I think that's a fine stopping point. We will start with the next chapter of material on Wednesday. Questions or concerns? Gonna go have a sandwich?